Okay. So before we start, let's see what are we gonna learn in this course. Okay. So today's lecture will be mainly about cell level. Okay. So when we talk about neuroscience, of course, you think about the whole body, right? But in the same time, you will have to zoom in into the cells, how the cells work, right? But the cell in this case is neurons, right? Which is the brain cell, right? So neuron. So we're gonna recall a little bit. This is maybe something you have learned in physiology. So we'll bring it back a little bit talking about cell neuroscience, okay? And then today we might have time to cover some of the system neuroscience as well. Okay, so you guys will we will talk about, some system in your body, right? So if you remember, we have, the system that control movement, system that make us, aware of the environment around ourselves, somatosensory system, those kind of system, and higher brain functions, which is how do you learn, how do you, memorize things, how do you dreams, those kind of things, right? And the case study would be some example of clinical problems, right, related to neuroscience. And then you guys will discuss about that example. Now, the last lecture, research in neuroscience, will be something that, interesting and it can be flexible. So I have what I prepared already, which is mainly about neurodegenerative diseases, some of the traumatic injury, some, for example, brain traumatic injury and then research related to that, or some of the research on spinal cord injury, for example, some of the neural stem cells research, for example, okay? So those are topics that I prepared for the last lecture. However, you guys can request, okay? So now it's a good time. So let's say I give you maybe until next week. If you want to learn something specific, okay? For example, I heard some of you mention neuro, psychology, right? So mental health issue, psychological problem, something like that, okay? I can add that into the last lecture as well, okay? I forgot to mention that. So my expertise, so when I did research, my main focus was in, the model for neurodegenerative diseases. I also did some research in schizophrenia patients as well. So anyone know schizophrenia?
Like
Yes.
hallucination. Uh,
Exactly. Yeah. Exactly. So yes, I did some of the research trying to looking for, biomarkers for those schizophrenia as well. So I can definitely talk about that. Okay. So some psychiatric or mental health problems at the end, right? A little bit at the end. And same thing, if you want any other, specific topics that you want to hear more, feel free, email me, okay? Send me, everyone has my email already, okay? So just send me an email, tell me, or you can type in the chat at the end or throughout this lecture also, I'll read it at the end, which topic that you want to hear, okay? And what do you expect to learn from the class? I would love to know that as well. Okay, you can type in the chat. Again, you can choose everyone. You can choose to send it to everyone, or you can choose just to send it to me, okay, in the chat box, right? So I can learn what do you want, and then I can, share that with you, okay? Because we have some limit time, so we can, choose what topic would be the best. Okay. Any questions in general about the topic? Oh, by the way, so the activity, so that would be related to. So I will send the detail of the schedule, I actually sent the schedule to Miss Yan. Am I pronouncing correctly? Miss Yan, right?
Yes.
Miss Yan. Okay, yeah. So I already sent the schedule to her. I don't know if you guys get the schedule.
Um, Usually if you send it to her, it will take a long time to reach to us.
Okay.
So
So can anyone like share this to your, I mean, your friend? Just
mm,
to emphasize, right, that this is the schedule. It's in a handout. It should be in a handout. Everyone should see, but just in case, so some of your friends might not know. So just want to make sure that everyone is in the same page, right? So we will meet Sunday. Next week we'll meet both Sunday and Monday. And then, the week after, 2 weeks after as well. And then at same time, 7 to 11. Thank you everyone for waking up at 7, by the way, on Sunday. All right. And um, so the time here, the reason why I have to do 7 to 11 because we have 12 hours different. Okay. And then I work, during the weekday. So now right now it's about almost 7:30 p.m. for me, my time. So the exam will be on April 19. Okay. Now, the detail for how are we going to evaluate in this course? So final exam, which is the April 19, right, will be 60%. Okay, so 60% will be the exam, and it will be multiple choice exam. Okay. Now, the rest 10% it's gonna be attendance. Okay, so basically, if you attend the course, you get like almost like 10% for free, right? So that's attendance. And we also have class, activity, and assignment. So together it's 30%. Okay. I will tell you for each activity in detail, but basically we're gonna have, some report from the case study, some group activity, and the final report as well. Okay. So those are, another easy 10%. So it should be relatively easy for you. Okay. Any questions about the detail of the course so far before we continue? Nope.
Um, Excuse me, ma'am, but I've just
Mm.
joined the class and have you done the attendance check yet?
Not yet. So I will. So the way that I will check attendance. So during the the course, I will send, I will do like a real-time quiz. Okay, it would be like easy quiz. And I will just send you the, short form, like Google form, to answer, short answer for the quiz questions, right? So that will automatically check attendance. Okay, so and I know that some of you might arrive late, or it's like 7 a.m. in the morning, so it's okay. So I will do that around, in the middle of the class. It's also check also if you like turn on, sign in and then go back to sleep or not. Okay. So make sure that if you really attend the class, you should not be, you should miss the quiz, should not miss the quiz. Okay. Sounds good. Any other question? Oh, TBL is team-based learning. So that will be some small group activity. Okay. And usually I will, assign who's in what group, who is in what group. But I think this year I don't know, maybe open for, random if you want to choose your own group. But I would need the name first. I would need like numbers, exact numbers, how many students, in total. But you can already, if you know that, who's you're going to work with, um, it would be nice to have, let's say, about 6 students per group. Okay, so if you can, form the group within your friends from now, and so you will be ready for that group activity. Okay, so I will ask to have like maybe around 6 person per group for those activity. All right, and we will talk about that in detail tomorrow. Any other question? Oh, I have another question. So do you have some kind of representative for the whole course or for each program? How does it work?
Each
So
program would have one representative.
Oh, okay. Can you maybe send me an email, each of you, so I know, so I have the direct contact. Just like introduce yourself in the email, like, oh, I'm a representative of this program. Okay, is that okay?
okay, I
All
can
right.
send the email.
Thank you. And then there will be 3 of you, right? If I understand correctly, 3 programs, correct? Or 2?
I think it's two.
2. Okay, 2 program. All right, perfect. Thank you. And so I can communicate with you, easier. All right. So if we don't have any other questions, so we can start. So I think now we have 46. Okay. And let's see how far I can go today. Also, because I just had a flu last week, and then my voice are a little bit, if I is talking for too long, it start to be gone, disappear. And so I try to like not speak too loud. I'm used to, shouting, like projecting my voice a lot, even though I'm in front of the computer. But anyway, let's hope that I can last, 4 hours today. Okay, so let's start. All right, so when we talk about neuroscience, okay, this is big, right? This is big term. It's the scientific study of nervous system. And of course, there are so many different branches of neuroscience, okay? So I think some of your some of the students that I teach before at Hanoi, when I went to Hanoi, they took my class, and then they were like, super interested in neuroscience, and then they apply for some of the scholarship in Europe, and now she's doing like PhD or something in Europe. And so I will be happy to write any, letter of recommendations for any of you who are interested in neuroscience program later. Okay, let's say if you want to learn more later, and if you find it interesting for you, and you want to apply to any programs, um in US or in Europe, in France, anywhere, okay, in Thailand, let me know. Okay, so again, like I say, a lot of branches in neuroscience, right? You can have the cellular neuroscience, where the neuroscientists in this field just really focus on neurons, right? How the neurons work, physiological property of each cell, right? Each neurons, right? How the neurons develop, how because those are underlying mechanisms, right? Those are basic information to make you know how it works. Even though nowadays we know a lot, it's still not 100%, right? The brain is like a black box somehow. We don't know exactly how it works like 100% yet. Okay, so everything is still um interesting and challenging, right? Neurophysiology specifically into, for example, if you look at action potentials, right? So those kind of things. Zoom in more into the functions. Clinical neuroscience, looking more for disorders, right? So cognitive neuroscience, understand nature of cognition. We will mention a little bit about these terms, right? Cognition is not only memory, right? It's also, how you aware of things, decision making, planning, thinking, memory, those kind of things is also considered cognition, right? So cognitive neuroscience is dedicated for study those kind of things and so many things, right? So on. So for today, for this course, we won't be able to cover everything, right? So we will talk a little bit, just, some of their cellular neuroscience because it's important basic background, right? And a little bit about the whole system neuroscience. As I mentioned in the last lecture, we will talk about degenerative neuroscience a little bit, right? How neurodegeneration occur, what type of neurodegenerations that you can see, the common types. Everyone know Alzheimer's disease, right? So we'll talk about Alzheimer's disease a little bit, right? We will also talk about some aspects of regenerative neuroscience as well, right? Regenerative neuroscience happen when you, for example, have some, injury, brain injury, spinal cord injury. In that case, can you repair or can the neurons be regenerated? Okay, so those will be something that we will cover. And like I said, again, any topics that you're interested, feel free to propose, okay, either through email or in the chat. Okay, so first fact, human brain is the most complex machine on earth, right? And if you know the numbers of the neurons in the brain, right? If you want to, so 100,000 million neurons, right? Huge amount of neurons, okay? More amazing than that, each neuron, so you have like, what, 100 billion neurons, right? 100 billion neurons. Each of these neurons sends the output to 10,000 neurons, okay? Thousands, or some of them send it to 10,000 neurons. We talk about each neurons, okay? Because if you remember, if you recall about the neuronal structures, you will know that it's branched a lot, right? It develop a huge amount of branch. So one neuron can send output to a lot of other neurons. In the same time, individual neurons, some of the neurons can receive input from so many other neurons, right? So 10,000 neurons in some cases, right? So it's a huge, complex brain, right? Everything is talking to, each other. So speed is also something very fascinating, right? So the neuronal signaling speeds vary from 0.6 meter per second to 120 meter per second, right? And you can imagine this, you think about your toe, right? You want to move your toe. The moment that you think about it, you immediately move your toe, right? So it's the proof of, how fast the neuronal signal travel, right? From your brain, that's where you think, right? And then whoop, your toe responds to it. That means the signal that comes from your brain down to your toe, it's super fast, right? Some of you might be super tall, right? Almost, some people almost like, I don't know, 200 meter. Same thing, right? They're thinking and then they can move the toe right away, right? So signal is very fast. And then we will talk about that also, how comes the signal travel that fast, right? So which is very important for, basic function and also the disease related as well, okay? So very important principle before we zoom into the cellular neuroscience. This can be the review for some of you that already took anatomy, neuroanatomy. So in the big picture, okay, when we talk about nervous system of the body, we have two major system, right? So two major system among several system. These two major systems are the first one, somatosensory system. Anyone remember what is somatosensory system? What is the function of somatosensory system?
I think we haven't learned about it, but based on the image, I guess it's receiving information from sensory tissue
Very
and
good.
process.
Thank you. Very good. So, yes. So you haven't learned these, right? Okay, that's fine. That's fine. It's good. See, like it's good that you tell me, so I know that. Oh, I assume that maybe you heard about this, but maybe not, or maybe you already forgot. So, somatosensory system, exactly like you said. So it's receive sensory from the body, okay? But more specifically, if you look at this diagram, right? It's receive the sensory or sensation from the body, okay? So we have, let's say, a receptor on the skin, right? We have the skin receptor that you can detect pain, temperature, touch, right? Touch or some pressure that are on your skin, right? We also have the ability to sense what's going on in our muscle and bone as well. Sometimes, a lot of time, you don't even know that these systems are working. For example, when you stand, when you run, okay, when you keep balance, right? So those receptors inside your muscle and bone and joint, they're working all the time to sense that pressure from your knees, from your leg, right, from your foot, and then send it up to your brain, okay? So the moment that you step on something sharp, like the sharp object, the reason why you feel pain is because you have the pain receptor on your skin, right? And that pain receptor is activated and it sends a signal up to the brain, okay? And because you have the areas of the brain that dedicate for that sensations, so you can interpret that, oh, that's the painful sensations, okay? Something stimulate your skins, right? So we will talk about these in detail in the, somatosensory systems in the next lecture. But here, I just want to show you that if you look at these tracks, right, each of these tracks, there's not one neuron, right? It's not only one neuron that go from skin up to, the final destinations in the brains, right? Those red dots right here represent the point of connections between different neurons, okay? In other words, we call these synapse, okay? Synapse mean when the two neurons are connected to each other or communicate to each other, okay? So you can see that there are a lot of neurons that are communicating to each other, right? And then finally, eventually send the signal to the brain area, okay? In this case, it's from the body up to the brain, okay? We also have some the spatial sensory system as well that detect sensation from eyes, from ears, right? Or from nose, right? But those systems are called spatial sensory system, okay? Which will be also in the next lecture, okay? So this is the sensory, right? Back from the body up to the brain. In contrast to the right side, it's the opposite, right? This motor system allow you to control movement. So it's the opposite way, right? So you have the centers that are located in your brain. And again, it sends a signal down to the muscle, okay? Anytime you make a move, anytime you move, whatever leg, fingers, arms, hand, you move the muscle. You stimulate the contraction of muscles in different parts of your body, okay? And the way to do that is that your brain have the center that send the signal down to the spinal cord, and then from the spinal cord, you send the signal down to the muscle to command that muscle for contractions, okay? For example, your toe, it has to travel long distance, right? From the brain down to the spinal cord, and then from spinal cord down to your toe. Okay. Again, same ideas. There are a lot of connections in between, right? All the red dots represent a lot of connections. Okay. Therefore, it's very important, first of all, to know how are they stimulated from the beginning, right? How are these receptors stimulated? How touch can, activate each of these receptors, and how those signals can travel through these long distance up to the brain. And then once it's in the brain, how do you interpret, for example? How do they make connections? Okay, so these are the questions that we will discuss in the first lecture in the cellular level. All right. Okay, so zoom in, right? Zoom in into the basic cellular level. So in your brain, this is the cells of the brain, right? And everybody knows that the brain cell is called neurons. Okay, so it's basic functional unit of the nervous system, neurons. And if you zoom in, so this is usually the cartoon, right, or model of one neurons. You have cell body, right? You have cell body, and then you have the axon, okay? Axon is the one that send out information, right? And you also have dendrite, right? Dendrite that you receive informations, okay? However, this is a cartoon, right? A very common cartoon, axon, dendrite. But in reality, you have these axons are more complicated than that. So this is, you can see from these pictures, right? So this is like complicated branching of the axon. This is the neurons in mouse brain, okay, that are labeled with fluorescence protein. So that's why you can see, the bright color, bright pictures right here. So you have a lot of branches in the axons and also dendrites, right? Branches everywhere. So keep in mind that your neurons are having huge amount of branches. And if you remember the first slide, we talk about connections, right? Input and output. That's why, one neuron can send signal to so many other neurons, and in the same time receive signals from so many other neurons as well. Okay, it's a huge complex. But when we talk about, when we learn, we have to simplify things, right? And that's why we zoom in into this simple model, okay? Cell body, again, cell body, or soma, cell. Cell body means cell body. And then you have axon, right? That send out information. And at the end of the axons, you have the connections, right? It connect to another neurons, right? And again, these connections is called synapse. Okay, this word is very important. Okay, so please remember this term, synapse. Why is it important? Because everything in the nervous system has to do with connections, right? So if you lost the connections, you lost information, okay? When you learn something new, what happened in your brain is that you are forming the new synapse, okay? Some cases, you make a very strong synapse. So the stronger the synapse you have, the better learning and memory you will have. Or the more synapse you have, the better learning you have. Sometimes this synapse can be disappeared. And that happens when you forget something, okay? Because those informations are not used. For example, when you don't use some informations, all the times, you maybe you see someone only once, right? And then you never meet them again. Those information, the first time you meet, maybe you form some connections, right? You form some new synapse in your brain. But then if you don't see them again, those connections are disappear. Okay, so that's why that's an underlying mechanism for learning and memory. And we will talk about that also a little bit in one of the lecture. Okay. Anyway, that's a synapse, right? So connections between the two neurons. More specifically, at the tips of the axons, through on the dendrite of the other neurons. Okay. Zoom in a little bit at the synapse. Okay, so this is where we zoom in, right? Again, synapse connections of the two neurons, right? And this is the tips or the end of the axon, right? We call this area presynaptic terminal. Okay, so this is a part of the axon terminal. Okay, presynaptic terminal. So what do you have inside presynaptic terminal? You have a lot of neurotransmitter, right? It's another important term that hopefully everyone know. Neurotransmitters, okay, which is the chemical molecules that you use to communicate between neurons, okay? And it's very important for activation or inhibition activity, okay, for these neurons on another neurons, okay? And there are several types of neurotransmitters depending on what kind of neurons you're talking about, what location of the brain, what kind of neurons. Some of the neurotransmitter can excite the next cells or next neurons. Some neurotransmitter can inhibit the next neurons, okay, or target cells, right? We will talk about that as well. Now, one thing to notice here, those neurotransmitters are mostly located inside the vesicle, okay, inside the vesicle called synaptic vesicle, okay? And then when this neuron is stimulated, you will release this neurotransmitter out from this vesicle, okay? Release it out, all right? Then on the other neurons, on the dendrite of other neurons, you call this area postsynaptic terminal, okay? So this is a synapse, right? And then on those postsynaptic terminal, you have receptors that will respond to neurotransmitters. All right, so make sure that you know these terms very well because it's the basic of, a super basic of the neurons, how they connect, okay? Zoom in a little bit on the pictures on the top right here. On the dendrite, if you zoom in on the dendrite, in reality, especially in your brain, on your dendrite, you have a special structure called spine, okay? Dendritic spines. So basically, if you imagine this is the, the long dendrite like here, you zoom in, you will actually see these mushroom shape structures here called dendritic spine, okay? And this is the areas that are enriched with receptors, okay? These areas have a lot of receptors. And again, number of these spine also determine learning and memory as well, okay? Also, very important for learning and memory as well, okay? Similar idea, when you learn something new, when something really significant, appear to you, or when you repeat, let's say you listen to this lecture 10 times, okay? This number of the spine will be increased, okay? And then you will learn that specific information much better and it will last longer, okay? Basically, you have more connections, all right? So just the simple terms on this slide, okay? Start talking about how does it work? So how does the simple functional unit work, okay? Again, this can be just the review for some of you who remember neurophysiology, right? Or it can be new for some of you, but it's very important basic information. So this is how the neurons is activated and then send information to the next neurons, okay? So basically, the property of the neurons are, first, each neurons can be activated, okay? And when it's activated, so when it's here, you're looking at two neurons right here, okay? The first neurons represent sensory neurons, right? Sensory neurons. So you can imagine that these can be the neurons that send the nerve through to your hand, for example. So this will be like the skin on your hand, right? And then the first neuron send the signal to the second neurons, okay? So you can imagine second neuron is located in the spinal cord, for example, okay, in the spinal cord. And then so on. The third neuron would be something that goes, that are in the brain. For example, the spinal cord send a signal to the third neuron in the brain, right? Similar to the pictures that we see before, right? Now, so the first property is that this neuron has the ability to be activated, right? And when the stimulus or stimuli activate the neurons, okay, for example, an activation can be different forms. It can be touch, right? It can be, chemical stimulations. It can be mechanical stimulations like touch, also pressure that push on your hand. It can be temperature as well, right? Cold or warm or hot. We will talk about the detail on these stimulus later, but this is in general terms, okay? But when the stimulus activate the neurons, your neurons will create the signal called action potential, okay? Now, we will zoom in into action potential in the next slides, but now let's look at the big picture. So the neurons zoom in, stimulate, right? When it reach the point called threshold, okay, the stimulus will change the property of the cell membrane, okay, until that potential of the cell membrane reach the threshold, and then you create the signal called action potential, okay. And why is it important? This action potential has the ability to travel along those long axons, okay? So one action potential develop, the second will come, and then so on. And then it will propagate or travel along this axon until it reach the end of the axons, right? So once it's reached the end of the axon, synapse occur, okay? Synapse, which is another term, synaptic transmissions, right? So this is the communication between the two neurons. So each of these we will zoom in and see what's going on, okay? So the first one, what is that peak right here? The shape that we see, action potential. So before understanding action potential, I have a questions for you. Anyone remember during the normal stage, before the neurons are, activate, before you stimulate your neurons, you have different kinds of ion in and outside, inside and outside of neurons, okay? So the amount of ion inside and outside are different. Do you remember, anyone remember what are the ion that are, high outside? Okay, I changed my questions, maybe easier. Sodium ion, it's high inside or outside of the neurons. Sodium, Na, sodium.
Is it high inside the neuron?
Sodium is high outside of the neuron. So outside is higher, okay? That's good. When you get the wrong answer, I like when students answer wrong, because you know why? Because you learn that much better. So like, oh, shoot, I got the wrong answer. And then you remember that answer, right? So now you know. So sodium is high outside. Okay, what about potassium? Potassium.
Then it would be high inside.
Exactly. So, and following that questions, what is the major reason that makes sodium higher outside? What makes sodium concentration higher outside? in resting, during resting, there is one pump that play major role to keep sodium concentration high outside of the cell. Anyone remember the name of that pump?
The sodium potassium pump?
Exactly, very good. So sodium potassium pump, or sodium potassium, another name, ATPs, right? And it got its name because sodium potassium ATPs, because it need ATP, right? It's an active transport, right? So you need ATP in order to pump sodium out, right? So this pump will maintain the membrane potential, which is the difference between the voltage, right, of the membrane, right? So that's why this pump three sodium out and pump potassium in exchange with potassium, two potassium inside the cell. Okay, that's why you have a lot of potassium inside the cell and then sodium outside the cell during what we call resting state. Okay, so resting level. All right. So that's why during the resting level, so more specifically, we call that membrane potential resting membrane potential, right? That's why there's a long explanation for these minus 60. Okay, I won't go there, but you can see that minus 60 during resting membrane potential is negative, right? So inside is more negative relative to the outside of the neurons. Okay, now I just want you to picture and visualize that, oh, the sodiums are waiting outside of the neurons, right? During resting, there are more sodium outside. So if you imagine there's a door or channel, right, on the membrane, and if this door is open, and if this door is the door for sodium, once it's open, sodium will flow in or out of the cell.
It will flow in.
It will flow in, right? Correct. So that's what happened when the stimuli hit the cells, okay? For example, if you think about your hand, right? And then your hand has the nerve that are underneath the skin, right? When you put the pressure on your hand, that's an example of mechanical stimuli, okay? So that mechanical stimuli will somehow open the channel, okay? Open the sodium channel because of the stretching of the, surface of the nerve, right? From that mechanical pressure. And when that happens, you can see that some of the sodium will come in, right? So increasing that number, right? Membrane potential. So from minus 60, you will start to increase a little bit here, right? And then once your membrane potential start to be increased until you reach one level, another door for sodium is open, okay? And when that door is open, sodium that are waiting outside, right, flow into the cells quickly, right? So that's why, and sodium has positive or negative charge.
Positive
Positive
charge.
Positive charge, right? So you can imagine easily that, oh, that positive charge is coming into the cells quickly. So that's why you have these shooting right here, right? And then you can see membrane potential from negative, it become more and more and more and more and more positive, right? So this is a result of sodium coming into the cells, right? So this phase of action potential is called depolarization, okay? So this is when the sodium coming into the cells quickly, right? Until at one point, that door is closed, okay? The door for potassium is closed, right? And then imagine another door open at this point. When the membrane potential reach the specific value, the sodium door is closed and the door for potassium is open. Okay. Now remember my questions. Potassium is high inside or outside?
It's high inside.
Inside, right? So when the door is open, potassium will flow from where to where?
Will flow from inside to outside.
Exactly, right? So again, so now in this case, only potassium will flow, right? And potassium also have positive charge, right? So when it flow outside the cell, right? So then membrane potential drops, right? Become less positive. You call these phase repolarizations, okay? And then a little bit happened after because the sodium keep leaking, keep going out, right? Because the door is kind of slowly closed. And that's why you have a little bit of bump here called after hyperpolarizations, right? Happening from the closing of potassium slowly, right? So that's why it's a little bit negative right here. But the key message, why am I showing this picture, is that those action potential or signal that you see, oh, sorry, those action potential that you see in here is actually a result of movement between ion in and out of the neurons. Okay, isn't it amazing? Right? Just two iron, right? Two major ion can create those signals in your neurons. Okay. And the door, I keep mentioning the door, those door, or more specifically, ion channel is very important. And you will see later, because if you use the drugs or some kind of toxin that block that door, okay, then you get rid of these signals, right? So you cannot develop this action potential. Then you block the signals in your neurons. Then you block sensations, right? Then you stop your neurons from sensing, the touch or pain or whatever. So that's the underlying mechanism for some of the drugs. Same thing, if you close that door, if you block that door for motor neurons, for example, then the neurons cannot command the the muscle. What happened after that? You can have like weakness or paralysis, right? So these little signals are very important, okay? So zoom in. We talk about that already, but this is just, show you more to be, correlated with the iron that we talk about, right? So again, during resting state, the channels are basically closed, okay? Both sodium channel or the door, right? Sodium door or sodium channel, and the potassium channel is closed, right? And like we said, sodium is high outside, right? And potassium is high inside of the cell. So this is outside, this is inside, right? This is a membrane of neurons, okay? Resting state, these channels are closed. And more specifically, this is V in this case is voltage gated, sodium or voltage gated potassium channel, okay? So when the door closed, nothing moved, okay? But when you have the stimuli, those stimuli will open this sodium channel, right? So sodium flow in, causing these depolarizations, okay? At one point, the sodium channel is closed, repolarization occur, right? Which is this state where the potassium channel open instead, and then potassium move from inside to outside, causing the membrane potential to decrease, right? And this is repolarizations, okay? Hyperpolarization occur from, if you look at the potassium channel here, it still remain open, right? Because it's kind of slow, right? So that's why some of the potassium still go out from the cells, and then membrane potential become even more negative, right? Until it go back to normal, which is the resting state, meaning that every channels are again closed, right? So you go back into the resting state, okay? Why are we talking about this? So again, now here, make sure that you know when you look at action potentials, right? And you know that what caused this depolarization and what caused repolarizations. And why is it important? This tiny channel, why do we have to talk about it? So, anyone know this fish? Puffer fish? Anyone have tried, anyone ate it before? Is it edible? Can you eat it?
They said that if the chef is very skilled, then maybe it's edible.
Yes, it's true, it's true, it's true. Where did you hear that from?
I don't know.
So, uh-huh,
Excuse me. Uh,
yeah.
um, yeah. Actually, the toxin of the pufferfish comes from the food. So uh, it's a certain kind of seaweed. So if you manage, if you farm them without using without in the habitat as there's no there doesn't have that kind of seaweed, then the pufferfish will not have toxin.
Okay. So there are another type of plankton that have similar toxin to the tetrodotoxin, which we will also talk about that as well. It's like one of the game that I will have it for you. But actually, you're right partially. So the toxin comes from the intestine of the pufferfish. Okay. So both of you are right. And this is very famous actually in Japan because it's kind of challenging, you know. So if the chef are certified, that means if he knows how to remove that organ, the internal organ out without, breaking those intestine, without breaking the internal organ, and then contaminate the meat, then you can eat the meat of the fish, right? But if you somehow damage that organ, the internal organ of the fish, then the toxin that are in that internal organ will contaminate the meat, right? And then when you eat the meat, some people still have some numbness feeling on the tongue, which they love it, okay? But if you, let's say, eat the whole fish, okay, what's gonna happen? Anyone have heard? What can happen if you eat like the whole fish? And if
Would it
Mhm.
lethal?
Mm. It can be. So it can range depend on how much toxin that you consume, right? If you consume just a little bit, maybe you will just have the, numbness sensations, right? On the tongue, the mouth, for example. But if the toxin go in your system, right? So all the nerve will be blocked, right? And what is it blocking, by the way? It blocked this sodium channel that we just talked about, right? That voltage gated sodium channel. So when this sodium channel is blocked, you cannot develop action potential, right? And remember, action potential is a signal that your neurons need, right? To send throughout the neurons to activate muscles, right? And everything. So when this sodium channel is blocked, you cannot do that, right? So you can have symptoms range from loss of sensation or weakness or loss of muscle movement, right? Or paralysis, right? And anyone know what's the cause of death? What actually caused death in the people that consume like high level of tetrodotoxin? If you think about muscle, what muscle that if it doesn't work, you die?
I think they would die of suffocation.
Say again.
Suffocation.
Yeah, more specifically, respiratory muscle, right? If you remember, right, you need some respiratory muscle to help, when you're breathing, right? So if that muscle is blocked, you can't breathe and then you die, right? So that's the most, cause of death in the patients that consume high amount of tetrodotoxins. Okay. So that's an example of how, important of these channels, right? And it can also be useful too, the knowledge. So local anesthetic, right? So anyone, I don't know if any of you have, experienced the use of local anesthetic. It's been used a lot in the clinic, also with the cosmetic as well, right? So before any, like surgical, skin surgery, any cosmetic surgery, um, or any small surgery, you apply local anesthetic, right? And so you block sensations, you block pain, right? So you don't feel any pain. And the mechanism is the same. So you decide the medicine or the drugs that block this sodium channel. But in that case, you block it, on the surface, right? Locally. That's why the drug is not going into the system, so it's not developing those systemic effect, right? So it's just blocking the pain, the sensation of pain, because it's blocked the sodium channel, right? So this is principle of how the action potential is developed and why these channels are so important. Okay, so just a little bit. So now after the action potential is developed, again, it can travel along the axon, right? And then arrive at the final destinations, okay? Or the second neurons, okay? So now what happened at the end of these axon, and what happened? How this first neuron can stimulate the second neurons. So this process is called synaptic transmission. Okay, so again, before we talk about that, remind you of the terms, right? So again, the tips of the axon, the end of this axon, we call presynaptic terminal, right? Because at this area, we call synapse, right? So this is presynaptic terminal. Again, inside, you have a lot of neurotransmitters that are packed inside the vesicle, okay? And this is postsynaptic cells. What do you have on the postsynaptic cells are a lot of receptors, right, for the neurotransmitters. So what happened is first you have those action potential that arrive at the axon terminal, right? So action potential propagate or travel through the end of axons. The important things first is that at the end of this axon, you have another channel. Okay, so previously for action potential, we talk about sodium, we talk about potassium, right? Now at the axon terminal, there is calcium channel. Okay, also very important because when the action potential travel to the tips of the axon, it will open the calcium channel. All right, and usually you have a lot of calcium outside. When the calcium channel is open, calcium will flow into the axon. Okay, and what happened inside this axon is that calcium will do something with this vesicle, okay? Will basically pull this vesicle to the membrane of the axon and then cause it to release neurotransmitter. So imagine if you block this calcium channel, what's going to happen?
Then the neurons cannot communicate to each other.
Exactly, because the calcium cannot flow in, vesicle cannot be fused with the membrane, neurotransmitter cannot be released, right? So this guy, the presynaptic neuron, cannot activate the second neurons, right? So it's very important. So yes, do you have anyone have questions?
Professor, was the vesicle have some chemical interaction with the calcium ion, so that you said that it have the calcium will pull the vesicle into the part that connect with the other neuron. So do they have any chemical interaction or something like that?
Yes, good question. So that's something that I'm gonna talk about in these slides. So basically, this is a zoom in, okay? On the left right here, this is like super detailed event, okay? Super detailed event where, you have the vesicle and then you bring it into the membrane, and then at the end you release neurotransmitters, right? But then look on the right here. So this is the zoom in of individual vesicle, okay? So this is the vesicle, and then inside the vesicle you have neurotransmitters, right? Okay, everyone follow me? So we are zooming in, okay? Basically, we kind of crop this area right here on the left here, and then we zoom in. So in the beginning, vesicle is floating inside, right? And then you have a lot of neurotransmitter inside this vesicle. On that vesicle, and here, what is this here? So this is the membrane of presynaptic neurons. Okay, so it's like this membrane. You can imagine it's like membrane right here. Membrane of the presynaptic neurons, right? And this is the vesicle, okay? In the beginning, vesicle is floating. So I'm gonna turn on my camera so you can see. Can you see me in the same time?
Yes.
Yes.
Okay, so basically you have the vesicle floating, right? And then you have the membrane right here. And usually on the vesicle, you have the proteins. So imagine you have these proteins right here on the vesicle. And then you have another proteins on the membrane, okay? So usually without calcium, vesicle would just float. And then these protein, let's say it will not interact to each other, okay? It would just like, okay, I'm gonna float, and then it will not attach to each other. When you have the calcium inside, you change the conformation of the proteins. And when that happens, now that protein can attach to the proteins on the membrane. Okay, so this is the way of, how you pull that vesicle to the membrane. And then once this vesicle come and then attach to the membrane, then the vesicle can open and release the neurotransmitter out to the synapse. Okay, so that protein is, if you look in the pictures, right, those are complex of the proteins right here, right? So basically with the calcium, so in the beginning, calcium channel is closed, right? No calcium come in, the vesicle is floating. When the calcium comes in, it changes the conformation of these proteins, and then it's pulling this vesicle to the membrane. And then this neurotransmitter inside the vesicle can be released out. Okay, we call this step vesicle fusions. So basically, calcium will change conformation of the membrane of the proteins on the vesicle, causing vesicle fusion to the membrane and release the neurotransmitter. Okay, so this is like amazing mechanism. And why is it important? Believe it or not, you know it. Oh, I don't have let's see if I have picture right here. No, I don't have it. But, okay. Everyone know Botox. You know Botox?
Yes.
What do you use for Botox?
It's used in cosmetic
In
to
cosmetic.
relax the skin.
Exactly. What else? Anyone else know how to use Botox?
Assassination.
Huh? Say again.
Assassination.
I'm not sure if I understand. Well, you use the Botox for the cosmetics, right? And you also use it in some kind of patients. When the patients have like spasm, right? When the muscle contract too much and you want to relax that muscle, you can use Botox as well. Some kind of headache, some kind of migraines, that are caused from too much contractions of the muscle, Botox can also be helpful. So actually several applications, right? But the major mechanism of the Botox, imagine, the mechanism, that's why it's used for the cosmetics. It's because it makes the muscle nice and relaxed, right? So if when you have the wrinkle on your face, for example, a lot of times it's caused from, that muscle that is like contract. So if you make it like relax, right? Relax all the times. So then you have, nice and, young face, right? So basically the idea is to make the muscle relax, right? So how do you make muscle relax? The mechanism of the Botox is that it break or it cut this protein complex. Okay, for example, the major one that we use nowadays cut this SNAP25 protein. So now if you imagine there's no more machinery, okay, that you cleave these proteins that used to fuse the vesicle, right, neurotransmitter to fuse to the membrane. The vesicle cannot be fused, so neurotransmitter cannot be released, okay? And now you cannot release neurotransmitters, so the nerve cannot send the signal to the muscle. And then as a result, muscle cannot be contract, right? So that's the mechanism. So basically you block synaptic transmissions, okay? So Botox cleaving these proteins, right? Blocking the vesicle fusions and blocking synaptic neurotransmitter release, block synaptic transmissions, okay? Same thing with some of the toxin that if you block calcium channel, for example, same thing happen, right? So no calcium channel, no calcium can come in, and then you block neurotransmitter release, okay? I think I have some video related to that later. We could come back later, okay? All right, so I'm gonna take a little break here, and then we'll come back to these slides, okay? Maybe, how long do you need? 10 minutes? Or 15 minutes?
It's up to you.
Okay, so let's say 10 minutes, all right? Then we'll come back. Okay, I'll see you. What time is it now? Let's say 8:35. Let's come back at 8:50. All right. If you have any questions, you can type into the chat box right now, okay? And I'll come back and I'll answer that. Okay.
This length of axon can be likened to a piece of fuse. The burning match under the axon represents an electrical event that generates an action potential. This event normally takes place at the axon hillock. Recall that to trigger an action potential, the electrical event must depolarize the axonal membrane beyond its threshold. That is, the membrane potential has to become positive enough to initiate the action potential, which is an all-or-nothing event. Once the membrane reaches that threshold, voltage-gated sodium channels open up, and sodium rushes in, as reflected in the upward deflection of the membrane potential. But how is the action potential propagated down the axon? Look at the area just ahead of where the action potential has just fired. It is becoming more depolarized. A millisecond later, the positive charge associated with the first action potential has spread to an adjacent region of the axon. This area reaches its threshold, and this segment of axon fires an action potential. The area of axon that fired an action potential a millisecond ago cannot become depolarized now. It is refractory because its sodium channels are inactivated. In this way, the action potential propagates in only one direction, like a fuse. Now, 2 milliseconds later, the positive charge spreads to the next adjacent region of the axon, and it reaches its threshold and fires an action potential. The region of axon that fired a millisecond ago cannot be depolarized. It is refractory. The area of the membrane that fired 2 milliseconds ago has now returned to its resting potential, but it is too far away to be influenced by the current action potential. In this way, the action potential migrates, like the fire on a fuse, toward the axon terminal, where it is delivered to another neuron. Now let's watch the whole process at high speed.
Okay. Yeah, I just think that it's like a clear, picture into how action potential move from one place to another place, right? So basically, you have the diffusions of those ions, right, along the axon and then trigger the action potential in the next areas. Okay, so a little bit of detail on that. Now, another video that I forgot to show you is really cool. I love it. So let's see. Okay. Can you hear the music?
No, we didn't hear anything from the video.
No, that's the fun part. Sorry, let me find out actually because I have so many video. Hold on. Audio. Ah, speaker. Mm, let's try. Wait, hold on. Oh. Wait, hold on. Mm, okay, let's try again. Make sure you can hear because I want to make sure because I will have a lot of can you hear now?
No, we still
No.
cannot.
Mm. Still no sound, right? Just picture, right?
Yes, just see the video.
Okay. Mm, let's try one more thing. Oh my gosh. Can you hear?
No. No, still no sound.
Yes. Okay, one more try, So background audio. Well, anyway, I'll work on it next time. Anyways, too bad you cannot hear it because the music is just very fun. Maybe you can look at the record, but anyone can tell me what's going on in the image, in the video that we just watched.
Like the cone snail, they sting the fish, and then the fish like get paralyzed, right? It cannot move, and then the cone snail just eat it like a.
Exactly. But what happened to the fish? How come the fish cannot move? Do you know what's in the corn snail? Poison. The corn snail has a toxin that block calcium channel. So, and the toxin is called conotoxin. And with that, when you block calcium channel, right? So again, when you block calcium channel, no calcium can come in, so vesicle cannot release neurotransmitters. So in this case, the muscle cannot be response, right? So cannot respond to the command because there's no neurotransmitters, right? So in the fish, so the fish is paralyzed, and we use that knowledge of the structure of the toxin from the corn snail to make some kind of the drugs that actually use in for the patients that have severe pain from the chemotherapy. Okay, if you search, you can look at, if you're interested, you can look at the chronotoxins and drugs, development or something like that. So this is another pictures that I just mentioned, Botox, right? So again, similar principle, but in this case, it doesn't directly block calcium channel, but it cut or damage or cleave the machinery to that fuse that play important roles in neurotransmitter vesicle fusion, right? So when it cannot be fused, it cannot release neurotransmitters. And so again, muscle stay nice and relaxed, okay? Another toxin that can come from snake, right? Or some of the plant is behaving similarly, right? So it's also block the calcium channel as well, specifically the channel for receptor called acetylcholine, right? So this is different in terms of locations, right? So in this case, previously we talked about presynaptic neurons, right? So the corn snail block the calcium channel on the presynaptic terminal. But in this case, on the postsynaptic side, you have acetylcholine receptors, right? For example, on the muscle. And so if you block these receptors, so usually you have iron, which is like sodium ion, right? Which can flow into the next cells and then activate the next cells. But if you, okay, I think I forgot to mention last step actually after we break. Okay, go back a little bit after the neurotransmitter is released. So you will see here after neurotransmitter is released. So in green here represent neurotransmitter that used to be in the vesicle before, right? But then after the vesicle fuse and release neurotransmitter, what happened in the postsynaptic cells is that the neurotransmitter can then bind to the receptors, okay? And it depends on what neurotransmitters and what cells you're talking about. If it's muscle, for example, that means this neuron directly control that muscle, right? Or it can be neurons to neuron, like in the brain, for example. Okay. So if it's muscle, the neurotransmitters that are used in muscle is acetylcholine. Okay, so ACH is one of major neurotransmitter that used in the muscle, right? So then that's it's linked to these receptors right here. Now this is acetylcholine receptor or ACH receptors, right? So you can imagine this is the muscle cells. So the membrane of the muscle cell will express these receptor, right? And then when the neurotransmitter bind to that receptor, it will open. So this receptor is acting as a channel, okay? But without neurotransmitter, the channel is closed. But when you have neurotransmitters that are released from the neurons, the channel can be opened, right? And once the channel is open, sodium can come in, okay? And then activate this muscle, right? So that's how the neurons send the neurotransmitter into another cells or the target organ and then activate the next neuron or activate the target cells, right? Now, if you block these receptors, then the sodium cannot come in, right? So that's happened, for example, in the neurotoxins from several kinds of the snakes, right? Including these snakes. I don't know if you have that in Vietnam, but I think in Thailand, we have some of these. So a very dangerous type of the snake, right? You also have another type of medicine that are developed from plant. So in the beginning, it's called the poison. It's a poison from plant that if you have ever watched some of the old movie and you can see like some in people who live in the jungle or stuff, they kind of use poison weapons, right? So they have like kind of a knife or sometimes the arrows, and then they coat it with some kind of the plant, and then that plant have some toxin called curare. So those toxins, similarly to the snake toxin, it block this acetylcholine receptors. So again, when it block acetylcholine receptor, even though you can release neurotransmitter, but those neurotransmitter cannot bind to the receptors, right? So it cannot activate muscle. So muscle become weak or paralyzed, right? So when you use this and then kill the animal, inject into the animal become paralyzed, and then you capture the animal, right? We use that knowledge to develop some drugs and then use these to block the acetylcholine receptor in some cases, right? Okay, so this is another short video, but I'll keep it for now because I can't figure out how to turn on the sound yet, so it will be maybe next time I will show it. Okay. So that's basically, general idea about how the cells, how the neurons are activated, how it's, transmit the signal to another cells, right? And you can see when you block some of the channels, those you have a lot of response, right? Before we talk about the system, with these slides, I have another actually short slides that we can take a look. Hold on a second. Okay, can you see the slide?
Yes, we can.
All right, so okay, so this slide, this is the picture of presynaptic neurons, right? Again, we talk about that presynaptic neuron. In this case, it has vesicle a lot of vesicle that have neurotransmitters inside. And in this case, the neurotransmitter inside is acetylcholine, right? And you can guess that down here are muscle cells. Okay, so this is the neurons that we call these motor neurons, right? So it's the neurons that will control these contraction of these muscle cells, right? So on the muscle cells, you have the receptors, right? Again, what kind of receptors? More specifically.
Acetylcholine receptor.
Exactly. So acetylcholine receptors, right? So these acetylcholine receptors, again, usually it's closed, right? But when you have action and potential trouble here, you will stimulate the vesicle fusion. You will open the calcium channel first, right? This is calcium channel, right? And then you will trigger the neurotransmitter release, right? So then blue dots right here represent acetylcholine. Then acetylcholine bind to the receptors, open the channel, and then allow sodium to come in, and then these muscle cells can be activated, okay? One other things that I did not mention yet previously is that imagine if you have a lot of acetylcholine releasing here, like if it keep releasing, what's going to happen to the muscle? If you have too much of acetylcholine releasing.
Maybe over contractions, like
Yeah.
so.
Over contractions, right? Or another terms is called spasm, right? Spasms, right? Muscle spasm. So you don't want that. And usually you won't have that because the body, the cells have mechanism on how to get rid of the excess amount of acetylcholine, okay? And the way to do that is that we have an enzyme, okay, represent in yellow dot, the yellow structures right here. And the enzyme is called acetylcholine esterase. Okay, so whatever, and with S usually is enzymes, right? You can guess that. So acetylcholine esterase will basically decrease or break down the leftover acetylcholine. Okay, so usually you have like a good amount to activate the muscle, right? Whatever that you have left that you don't use, it will be destroyed by this enzyme. Okay, so it will destroy acetylcholine. And so preventing too much contraction of the muscle. Okay, so this is what happened, right? So this channel right here, what kind of channel right here? If you guess. It's like the channel that express along the axon. Sodium channel, right? So this represents sodium channel, okay? And then this one represents potassium channel, okay? Which is actually expressed, along the axon. But this picture is kind of like, shrink everything. So that's why you see it here, but it's actually expressed along the axon. But what we're gonna do here, I'm gonna show you. So A, B, C, D right here are the names of the toxin, okay? And some of these you might have heard already, some maybe not, right? So we will guess. The questions are, what does this toxin come from, right? For example, let's start with the first one. This one. Anyone? Which one that it comes from? Should be easy because we talked about it already. You can type in the chat also, so you can kind of like participate a little. You can just type in like A, B, C, D. So the toxin in the picture basically provide botulinum toxins, right? Exactly. So this is the botulinum toxin. So basically originally when we discover botulinum toxin is actually a toxin that cause, death. And the reason for that is because it's actually first found in when people consume some of the homemade preserved vegetable or food. So the toxin comes from bacteria, okay? And the bacteria grow, in those kind of like, dirty, not clean, preserved food, right? And so the patients ingest those toxins in a high amount and it goes systemically and, similar to tetrodotoxin, but different mechanisms, right? So the people die from that. But then later they just, invest the specific type of the botulinum toxins and then they inject it locally. So you want to make sure that when if you know somebody that want to get Botox, make sure that it's really local, right? Because you don't want it to like float in your bloodstream and then block every muscle in your body, right? You don't want that. You want it to be like localized only in the facial muscle, okay? So, and again, remember what is the mechanism of botulinum toxin again, anyone?
They cut the protein.
They cut the protein that play a role in vesicle fusions, right? So basically they prevent synaptic vesicle fusion and block neurotransmitter release, right? Or block synaptic transmissions, right? It's very good. All right, next. So Clostridium botulinum is the name of the bacteria that produce botulinum toxin. What about this guy? This one also we talk about it. Which one? C, right? Conotoxins, right? Conotoxin, cone snail. And what the mechanism again?
It's block the calcium channel.
Exactly. It's block calcium channel at the presynaptic terminal, right? So no calcium. Again, no calcium, the vesicle fusion cannot be occur and you cannot release neurotransmitter. This guy. Tetrodotoxin, right? Yes, tetrodotoxin, pufferfish, right? Isn't it cute? Right? So tetrodotoxin, what is the mechanism again?
It
Is
it
it
blocks sodium
it's
channel.
no.
Block sodium channel and then therefore block action potential, right? So the action potential cannot be created, right? So you cannot activate the nerve or muscle. This one, I guess. Have you seen this in Vietnam? Hopefully not. Anyone wanna just make a guess?
D.
Actually, H. Okay. This one is it's called banded krait. It's very poisonous snake, right? And the toxin is the bungaroo toxin. Okay. Similar to, the previous picture that I showed you earlier, it block acetylcholine receptors. So when it's block acetylcholine receptors, even though you have neurotransmitter release, you have neurotransmitter floating here, but you cannot activate the receptors, right? Because this one is blocked. Okay. So similar symptoms, you have the paralysis and death. This one, we talked about it already. Cura, right? Or tuberculines, that's like a specific names, right? So again, block acetylcholine receptor. This one, this one is interesting. And guess, no, nope, not E. Almost there. We only have a few left. Nope, not J. B, yes. So anyone know what are we looking at? Oh, I already put the answer. Anyway, so this is we are looking at the plankton. Okay, so some kind of plankton in the oceans are have different colors, right? In this case, it's like red, orange color. So basically, these planktons are poison. So it has the toxin called saxitoxin. Similar mechanism as tetrodotoxin, very similar, okay? Blocking sodium channel. So what happened is that the shellfish eat this plankton. And then if the human consume the shellfish, then they get these toxins, okay? There was some incident somewhere in US, I don't remember exactly, but the whole village basically have some, problems. They have like some kind of like weakness or some systemic problems, almost like paralysis, and it's called paralytic shellfish poisoning because they consume these shellfish, right? And then these plankton come in, specific time, season, so they don't know that, and they just consume the shellfish that eat these plankton. So again, when they consume that in a high amount, so it's blocked sodium channel, right? So you cannot develop action potential. And so many symptoms can develop. Worst case, paralysis, right? Okay, mamba snake. What else do we have left? The dendrotoxins, right? Another toxins that block the interfere potassium channel. This one, good guess, or if you know, good job. So this is basically the plant that is the source of what called physostigmine. Okay, so basically this physostigmine will inhibit the function of the enzyme that break down acetylcholine. Okay, again, to remind you, we have an enzyme, right, called acetylcholine esterase that will usually destroy or get rid of the excess amount of acetylcholine, right? Now imagine if you have this physostigmine, okay, which block the function of these enzymes. What's going to happen to acetylcholine and the muscle?
So I think the acetylcholine will accumulate and stain the receptor and will cause muscle spasm.
Exactly, right? So if you have, toxins from these plants and then it's block this enzyme, so you're gonna have high amount of acetylcholine, and then it will cause muscle contractions, right? However, so that's the principle. Very good. Humans are very smart, okay? There is a disease, right, that you might have heard or not. But one of the disease caused by abnormal antibody in your body is the autoimmune disease. So basically, the patients will have antibody that will destroy their acetylcholine receptors. Okay, so as a result, the muscle responds less to acetylcholine because it doesn't have receptors, right? So in that case, you want to increase the amount of acetylcholine in order to compensate with the receptors that are destroyed. So how do you increase the amount of acetylcholine right here? And we know that, oh, there's this compound that can eventually will block this enzyme, right? So when you block this enzyme doesn't destroy acetylcholine anymore. So you can have more acetylcholine to function at this area. Therefore, we use the knowledge of this physostigmine and we create the new medicine, okay, called, one of them is, for example, neostigmine, for example. And we intentionally use that to block the function of acetylcholine esterase in order to increase acetylcholine, in order to help the patients, right? Some of the patients that have autoimmune disease that have less amount of the receptors, for example. Okay, this is just additional things, right? I'm not gonna like test you in the exam for this one, but just to, something interesting to know. Okay, all right. Now, last one, since it's the last choice, so it's the J, right? So this is similar idea, DFP, right? Or it's the compound example of the compound that strongly, same target, block acetylcholine esterase, okay? In this case, it's by very tightly. That means it's basically destroy the acetylcholinesterase functions, right? And this is found in like pesticide, insecticide, for example. It also found in the nerve gas, okay, that people use in some kind of war, right? Nerve gas. And again, if it block the acetylcholine esterase, it increased acetylcholine release, right, amount in here, right, and eventually cause muscle spasm. And what happened when you have the spasm for a long time? At the end, that can lead to muscle weakness as well, because basically you run out of all acetylcholine, right? You kind of like stimulate, and at the end you end up with, first start with spasm, and at the end you have the weakness or even paralysis as well. Okay, so this is just an example of like, oh, we haven't finished yet. This one, what is this one? Pretty looking. This is tetanus toxin. Okay, similar target to botulinum toxin, but the mechanism of action is a little bit more complicated. Okay, so I won't go there too much. All right, so just example of, this little area right here at the end of synapse. This is just synaptic transmission. There's so many things that, can block it, right? Either the toxin that can cause death, can cause paralysis, or some of the medicine can be developed, right, using this knowledge as well. Okay, so just to make you see why do we mention about that. Okay, now I'm gonna switch to back to this. Hold on a second, let me check something. Give me 2 minutes, okay? Okay. All right. Mm, okay, where were we? All right. So, okay, so next we have some time. So we will move on to a little bit into the, system neuroscience, where we will talk about, each of the system, the somatosensory system, motor system, how do we sense, the environment, how we react to the environment, right? Using those two systems. And then spatial sensory system, how do we see, we hear, we smell, those kind of things. And so, and also a little bit, maybe the third lecture also, we will zoom in about, the higher function of the brain, which is the sleep, memories, those kind of things, okay? And again, emphasize, so for those of you who arrive later, maybe, don't forget to, if you want to learn something, specific, like you have a specific topics in mind that you want to learn, like, oh, I came across this kind of research and I want to hear more about it, feel free to suggest that either in chat here or email me, okay? If I have enough time, I will insert that in my last lecture for you, okay? Now, before we move on, so this is how I'm going to check your attendance, okay? So I'm gonna put this so we can, this is one question quiz. I'm gonna put this in the chat box so you can copy. Okay, hold on a second. Let's see if it goes. Okay. Wait a minute. Okay. Oh, I don't want a screenshot. Anyway, can you get it? Can you open the form? Anyone?
Yes, yes.
Okay.
They said that we need to
we need
request for
permission
access.
to, yeah.
Can you open the access of the phone?
Okay, let's see. I see it's like the first time, so everyone can, who else that hasn't get the access? Let's see. Can you access now or still?
Yeah, we can access now.
Okay, anyone cannot access?
I still can.
Me, I still can.
Okay, so I think,
Cannot.
okay. I think there's, it's good. This is the test basically anyway. So I will have to work on it to allow everyone to access. Maybe there's something that I click and then, block the access. Anyway, don't worry. So this is just a test. Okay. Next time I'll work on it and then make sure next time everyone can access. All right. Yeah, I don't know how, but I will find out. Okay. So anyway, I will capture the screen for you this time so you can see the questions. It will look like this. Hold on a second. Okay. So it will be something like this, you know. You can answer me right now in a chat box. Okay. Can anyone answer in the chat box? It will be these type of questions, you know, like easy, short questions, one question. And then, but the most important thing that you have to do is that, yeah, very good. So you have to put your code, right? So this, and in this format, the code first, right? Code, and then your name, your last name. So this way, when I get your response, I can check for attendance. Make sense? So I would know, you know, this response, okay? And don't worry too much. I mean, try your best to answer the questions, but even you get it wrong, it's fine. So think about it as like formative quiz, formative exams, right? Formative questions, just to inform you that this is important, right? But most the purpose is actually to check the attendance. Okay, so it will randomly appear in the middle. Okay, so if you're a little bit late, don't worry. Usually, you know, if you pay attention throughout the lecture, you should be fine. Okay, all right. So for this time, again, it's like you know, I assume that everyone is here then, but then next time I will find out. Let me make a note before I forget. That everyone can. Okay, two things now. First thing is audio that I have to make sure. Second is the quiz. Okay. All right. Any questions before we move on to the next system? Any questions related to this cell level? Oh, now you don't have to worry about, you know, telling me name or whatever, because this time, again, like I said, it's not count. I don't count the attendance, okay? I assume everyone is here. So I will start, but I will start counting next time, okay? So, any question? All right, if not, then let's move on. Okay, stop share. And then you should have the second handout that I sent you. Actually, let's take give me another 10-minute break, okay? Let me pull out some of the information. Maybe like 12 minutes. I will see you, no, 12 minutes will be 55, 9:55, okay? And again, if you guys have any questions, type in the chat and I'll come back and answer that, right? Okay. I changed something. Let's try again. See if you can hear the audio. Yes? No?
No, we still.
Okay. Never mind. Okay, I need more time. Okay. What about, and I changed something in the quiz, so I'm gonna paste again here. Okay, hold on. Dun-dun. Okay, let's try again for the quiz, but I won't take I won't take it this time, but just to try if you can access now. I put the link in the chat. Can you access now? Anyone? Okay, perfect.
Yeah.
Good, 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 good. Okay. Okay, one thing done. Next audio. All right, so don't worry about that quiz now. Okay, so now it works, but again, this time I will not take that, but I will start next time. Okay, so make sure that you participate, and I will open it for about like 5 minutes. Okay, so make sure that you're paying attention the whole time. Okay, all right, so continue. So no questions from the previous part. Let's continue. Now, second part will be system. Okay, where am I? Okay. All right, good. Okay, so now in here, major topics for this one will be overall organization, right? So for those of you who already learned anatomy, this should be easy. Okay. Right. So the slides that are missing are, usually those are like special example, like the toxin and stuff. I can send you that later if you want. Yeah, but just, if it's not there, that means it's not like it's the key message. It's just like a special, example that I put it in, okay? So you don't have to worry about it. But if you like, I can send it to you also, the for example, the toxin stuff. Okay. All right. So again, this lecture we will talk about some basic information of each system, right? Somatosensory system, which is the system that we use to detect sensation from the body, right? Somato mean body, soma mean body, right? So how do we detect pain, detect temperature from skin, for example, or how do we detect pressure from bone and joint and muscle, for example. And then we can't only, sense the things around us, right? We have to respond. So motor system is the system that we use to respond to environment, right? To move the body. And then spatial sensory system. We might just finish the first one today and we will continue next week, right? For the somatosensory, for the other system. So again, overview for, just review basic information about the component of the nervous system, right? So we have two major system when we talk about nervous system. We have central nervous system, which include brain, and spinal cord. Okay, so this is central nervous system, brain and spinal cord. And it will be very important later. So you have to really appreciate and know that, okay, when we talk about the central nervous system, we mean brain and spinal cord. Okay, and there are some major difference that differentiate between central nervous system and peripheral nervous system, which is peripheral system include the nerve that are outside brain and spinal cord. Okay, so those include cranial nerves, right? Some of the nerves that are coming out from the brainstem, basically. We'll talk about these in detail. And some of the nerves and all the nerve actually that are coming out from the spinal cord, okay? We call that spinal nerves, which include motor nerve and sensory nerve, okay? So make sure that you know this basic principle, okay? Now we zoom into the first part, the brain, right? So there are different parts of the brain, right? This is like major, some basic information, right? In detail, you will divide the brain into like tiny different area. But in this case, it's like big major areas. So first of all, you have two sides, right, of your brains. We call that two hemisphere. We have left hemisphere and right hemisphere. Okay, and these two hemispheres are linked together by the huge tracts of axon. Okay, so you remember the axon is the part of the neurons that send information out, right? So these are bundle of, by the way, the axons, the neurons are usually stay together. The bundle of neurons are form nerves, right? Nerve bundles or nerve fiber. So when we say nerve fibers, it's make sure that you know that it's multiple neurons coming together. Okay. So this is a bundle of those nerves, specifically the axon that link between left and right hemisphere. Specific name for this tract, it's called corpus callosum. Corpus callosum. Okay. So if you cut this corpus callosum, you split the brain into two hemispheres. Okay. But usually you have these corpus callosum that link, so some information from left can transfer to the right, and then information from the right can transfer to the left side. Okay, so looking on the side here, you have the big brain called cerebrum or cerebral hemisphere. Okay, this big brain right here. And then the corpus callosum that linked, right, between the two brains, right? And then down below, you have what called the encephalon. We will zoom in a little bit. Then down here, you have the brain stem, which composed of three major parts, the midbrain, pons, and medulla, together, acting together, controlling a lot of functions in your body, right? For example, respiration, so many other things. Then the brainstem also linked the big brain or the cerebrum right here to the spinal cord. It's like a bridge, right, between big brain and then the spinal cord down here. Okay, so this is major part. The tiny areas, the tiny part of the brain on the back right here is cerebellum, right? And when you think about cerebellum, think about movement, okay, motor learning or movement or balance, for example, okay, cerebellum, or coordination of the movement, okay. All right, so the most important part that we will spend most of the time is the cerebral cortex, which is a big brain part of the brain right here, okay. So when you look at the cerebral cortex, this is from outside, right? So the front will be this side, right? This will be the front part. This is your back of the brain, okay? We divide the brain or cerebral cortex into major lobes. We have the frontal lobes, right? The front part. We have the occipital lobes on the back. We have parietal lobes on the in between frontal and occipital lobe in blue right here. We have temporal lobe on the side, right? And again, I won't go into like anatomy on like, okay, how do you separate each lobe and those lobe. I would focus more on function, okay, on each of these lobe. So anyway, and you can see, in detail right here, but we will go slowly one by one later, okay? For example, in the frontal lobe here, the very front part here is very important for higher functions, right? So this will be in, one of the lecture in 2 weeks when we talk about decision making, planning, some reasoning skills. There's something to do with our front part of the brain, okay? The regions in red right here is the regions that control movement or motor cortex, right? So it's the area that have the neurons that will send the track down to the spinal cord, and then from the spinal cord you have another set of neurons that go to muscle, right? And you can control movement of your body, right? So this is called motor cortex, right? So if you want to know any anatomy from this slide, one important thing here, you can see the big, kind of a groove in the middle, central, right? So this part is the part that separate between the motor cortex and the other area behind this groove is called the sensory cortex, okay? So you can imagine these areas control movement, but the area behind that, as its name, sensory cortex, receive information, right, from your body, from your skin, the bone, the joint, for example, different parts of your body, right? So this is the somatosensory cortex, okay? And on the back right here, occipital lobe or visual cortex, right, located in these areas. Again, receive, as its name, visual cortex, it receive information from the eyes, right? Play a role in vision, right? For hearing, you have one of the areas on the temporal lobe that detect or receive information from the ear, right? So we'll talk about that in special sensory system, okay? So those are just for now, just like big picture from the cerebral cortex, right? Big part of the brain. And then, so this will be the cerebral cortex. And then this, again, the corpus callosum. Zoom down to the diencephalon. This area composed of two major parts, thalamus and hypothalamus. So thalamus actually composed, in this picture, it looks like a big area right here, but in reality, it composed of so many groups of neurons together, cluster of huge amount of neurons together in this area. And it plays so many functions, okay? But one of the important functions that I want to, emphasize is that it's function as the gatekeeper. Basically, what does it mean, gatekeeper? So it's a processing station for all of the sensory information. What does it mean? For example, you have the sensation comes from the body, right? From your hand, from your leg. Those sensations send the signal, go up to the spinal cord, right? Up to the brainstem. And then before it reach the area of the brain, it has to transit at the thalamus first. Or in other words, it has to synapse with another neurons in the thalamus first before it go up into the somatosensory or sensory area, okay? Same thing with vision. Signal or nerve from your eyes, right? From your eyes, part of your eyes, the retina, back of your eyes, right? Will have a lot of nerves. When your eyes are activated by light, or when you look at the image, right? Those nerves will be activated and it will send a signal through the nerve, right? But that nerve doesn't go all the way to the cortex, right? We talk about the visual cortex at the end, right? But the nerve from the eye doesn't go all the way directly to the visual cortex. it will have to synapse with some neurons in thalamus also, right? And then neurons from thalamus can send the signal to the visual cortex. Okay, that's why we call it gatekeeper, right? Or transit station, you can think of that, right? Because all of the sensory informations have to go through thalamus, except olfactory, okay, smelling, right? Otherwise, vision, sensory, somatosensory, auditory, hearing, all the sensation have to go here first. Okay, so that's thalamus. And hypothalamus. So some areas below the thalamus, okay, also have groups of neurons that are located under the thalamus, okay? And these neurons are playing a role in so many things. Ew, my cat just makes some weird noise. Okay, so okay, so hypothalamus just play a role in so many things, right? Endocrine function, autonomic nervous system, homeostasis, right? So those are down here, okay? And, if you learn physiology, you probably remember hypothalamus, play a role a lot in endocrine system, okay? Then below that, so this is a group of hypothalamus, right? So you can see different functions of hypothalamus. And again, groups of neurons, right? Now below that is the brainstem, midbrain, pons, medulla, right? And you can see, different function as well to control your basic function of your body, right? Then you have down below that, you have spinal cord, right? Now, those of you who took anatomy already, you probably have to remember different section, right, of the spinal cords, right? But in general, the principle is that you have the spinal cord, which is basically the nerves, right? It's full of the nerve that either come down from the brain. So you have the brain, the cell body of the neurons that we talked about, right? Located on the brain. And then those cell body will send the axon down to the spinal cord, right? For example, those motor neurons, right? It will send the axon down to the spinal cord, okay? And then in the spinal cord, you will have another set of neurons or nerves that send out, motor neurons to the muscle, right? In the same time, spinal cord also have some nerve from the body, sensory nerve, right? And then that go up into the spinal cord and then go up to the areas of the brain that detect sensations, okay? Now, in the spinal cords, we divide them into several areas, okay? CTLS, right? Cervical, thorax, lumbar, sacral, right? So that's the little details divided into different areas, okay? If you cut each section of the spinal cord, you will see, the cross section right here, okay? And the spinal cord is, spinal nerves is supported by the disc, okay? And the vertebra, right? So zoom in into the nerve itself, right? The spinal nerve. And again, you will see several areas. So you will see the root. This is one side. You have the sensory root. And you can imagine sensory root meaning the nerve that come into the spinal cord, right? Come from your skin, for example, and then come into the spinal cord. Another side, you have motor root, right? Which is the nerve from the spinal cord that go out to muscle, right? So that's why in the spinal nerve, you have combinations of sensory root, sensory nerve, and the motor nerve, okay? And so this is just the zoom in picture, right? So you have the dorsal side and then the ventral side, right? So just a little bit of the terms, but don't need to remember in details, right? Again, I don't emphasize in anatomy too much. So again, to emphasize, you have the somatosensory. So this is a signal that come into the spinal cord, and it's both same between left and right, okay? And you also have motor nerve that go out to the muscle, okay? So again, key idea, spinal cord, you have the sensory nerve coming in, and then the motor nerve going out, right, for now. In addition, so those spinal cord, then again, central nervous system, we have brain and spinal cord, right? But then the nerve that are coming out is considered peripheral nerve, right? So which is all the nerve that are outside of here. It's considered peripheral nerve. Another type of peripheral nerves are nerve that coming out from mostly the brain, the areas of the brain stems, right? And those nerves are special nerves called cranial nerves, right? Some of those nerves, most of those nerves control, either sensory or movement of the face, right? For example, you have nerve 1, which is the olfactory sensory nerve, right? That's and red fibers, whatever representing red, are the sensory nerve. So cranial nerve have the mixture between sensory nerve and motor nerve. Okay, so you have sensory nerve, for example, this one, nerve number 1, send the signal from the nose, right, olfactory smell coming into your brains. And 2 is the optic nerve, which is again sensory nerve, bringing in information from your eyes, right, bringing into. You have the nerve 3 in blue here. That means what? That means it's the motor fibers, right? So nerve 3 can control, along with nerve 4, can control, the movements of your eyes, for example. Okay, so you don't have to remember any of these nerves. Okay, it's just to show you examples, right, of all. And we have the 12 cranial nerves in total. Okay, and you can take a look at these yourself later to see that, oh, you have so many different nerves that control different organs, right? Different areas of your face or organ in your body. One thing to pay attention, vagus nerve or nerve 10, right? This is the nerve that is very important to control the internal organs, right? Your intestines, a heart, lung, those internal organs are controlled by vagus nerve. Okay, so just back to the brain. So another part of the brain is the back right here, cerebellum, right? So cerebellum, again, when you think of cerebellum, you think of coordination of the movement. Okay, all right. Okay, give me 2 minutes. I forgot my water. I'll be back. Okay. All right, so let's continue. So now let's zoom in each system. Somatosensory system. So first system, again, when we get information from the body, right? Somatosensory system. And when you talk about, if you just say the word sensory system by itself, it will include somatosensory system and special sensory system, okay? In this case, we will start talking about somatosensory system first, which is, again, system that detects sensation from your body, okay? And in order to detect that sensation from your body, which means skin, again, skin, bone, joint, muscle, right? So you need 3 major components, right? First components are sensory receptors, okay? So those can be receptors on the skin, in the joint, in the muscle, for example, okay? So those receptors are required. And then second compartment or component is the nerve or the pathway, sensory pathway, okay? In here, represent with this long pathway right here. Those pathways, so the somatosensory pathways composed of the sensory neurons, the first neuron, okay, sensory neurons that in this example coming from the finger, right? Fingertips, right? In red right here, this is one example of the first neurons in red, right? First in red. This is represents cell body of the first neurons. Now the special property, special structure of the sensory neurons is that the cell body located here, this area is called ganglion, okay? And this area can, these cells have bipolar, which means it's have the two sides of the neurons coming out from the cell body right here, right? So it's a little bit unique compared to, other type of neurons. So when you see this, you can tell that, oh, this is the sensory neurons, okay? So one side will receive information from the receptor and another side will go into the spinal cord, okay? And then synapse or connect with the second order neurons, okay? Second order neurons. And then third, again, it will relay or transit or make a connection in thalamus, right? Remember the gatekeeper or transit stations, right? So synapse occur here. And then another, the last neurons going from thalamus and arrive to the final component, which is the brain center. Okay, the brain center in this system is called somatosensory cortex. Okay, so in order to sense any information, and then there's some details about the pathway. Some can go to the red pathway, some can go to the blue, right? We'll talk about that in a little bit. But same principle, 1, 2, 3. If you're missing any of these components, you can't sense, whatever you touch, the pain, or, you can't detect the pressure normally, right? So all of these component has to be intact. Because sometimes you can have, let's say, if you have problem with the pathway, if you have some problem with spinal cord, you can have problem with sensations, right? Or if the patients have stroke in the somatosensory cortex area, or have the brain injury, right? Have some trauma, some accident, and then bleeding in this area, and then this area is damaged, then they're gonna lose the ability to sense the sensation or stimuli from the body, right? So that's why these major components are important. Zoom in into each of the component. First component is sensory receptors, okay? And we characterize the receptors based on types of receptors, locations of the receptor, and types of receptors. So locations of the receptors can be classified by, for example, if it's outside on the skin, for example, we call that receptors exteroreceptor. Okay, mostly skin receptor because it's outside, right? Outer surface of your body, external receptor. And those receptors composed of the pain receptor, temperature receptor, touch or pressure receptors. Detect all these sensations, okay? The receptors that are located in the muscle, tendon, or joints together are called proprioceptors. Okay, proprioceptors. These receptors detect movement or joint positions. Okay, which makes sense, right? Because you use the muscle to move and joint position to basically move, also stand or move or jump or walk, right? Those pressures will be sensed by proprioceptors. If you have the receptors in the visceral organ, right? For example, in your gut, in your intestine, right? That receptor is called enteroreceptor. Okay, so enteroreceptors. Those can detect movement of the gut or even pH as well of the blood, right? So those are based on the locations, right? But you can also characterize receptors into what type of the stimuli that the receptor detect, right? For example, receptors that are receive information from chemical stimuli, we call chemoreceptors. Okay. Can anyone think of example where can you find chemoreceptors in your body?
Uh, it would it be inside the blood vessel to check the pH of the blood?
Yeah, you can have some of that. Yes, very good. What else? Anywhere else?
I think
In
maybe
the
in the
nose?
olfactory in the nose.
In the nose, right? Good, in the nose, because you smell something, right? So those are also considered chemoreceptors. What else? Some of you are probably using it right now for breakfast.
In
Huh?
your tongue, maybe in your tongue, you can
Your,
feel the taste of food.
yeah, your tongue, right? Your tongue. So you can feel, you can taste, delicious food, right? Breakfast in the morning, right? Okay, so that's a good example, right? And a lot of places chemoreceptor. Very good. What about photoreceptors? Easy. Eyes, right? Only in the eyes. Thermoreceptors on the skin, right? Most of the time. Mechanoreceptors. Where can you find mechanoreceptors?
Arm or leg?
Yeah, on your skin, right? On your skin, your joint, your muscles, right? So actually all of these proprioceptors are actually considered mechanoreceptors. Anywhere else other than those joint, muscle, tendon?
In the gut with
Yeah,
the
in the gut also,
movement.
yeah. In the gut, you also have the mechanoreceptor detect, movement. Anywhere else? This one is like a lot of people don't think that it's actually mechanoreceptor. It detect vibration.
Ear?
Yeah,
Yeah.
exactly. In your ear. So in your ear, the receptor in your ear is mechanoreceptor. And we will talk about that when we talk about the ear, sensation from the ear. What is nociceptor? Anyone? Anyone know what is nociceptor? Yeah, nociceptor is another name for pain receptor. Okay, so pain receptor is expressed almost like everywhere. Okay, so the pain, anything that damage the cells, right, can trigger or can activate pain receptor or nociceptor. Okay, so right, this is a major types of receptor in your body. Okay, then zoom in a little bit to the skin, for example. How many, well, like how many types of receptor we have in our skin? Like so many of them, right? So if you look at this picture, it's like a piece of the skin, right? This is the outer layer of the skin, and you have the hair root is here, and then underneath the layers of outer layer of the skin, you have so many other types of receptors, right? Some are on the surface, some are deep inside. Okay, first type of the receptor under the skin is the hair follicle receptors. Basically, this is one idea, one thing that I forgot to mention is that Usually these sensory receptors are the end of sensory nerve, right? That are change the structure or adapt and then have, space, it's form specific structures at the end, the tips of the nerve, okay? For example, here you have what called Meissner corpuscle, here you have Pacinian corpuscle. It's the special, structures, right? In this case, the hair follicle, it's the tips of the axon that wrap around the hair follicle, okay? So if you imagine, if you move, if this hair on your skin move, it's gonna trigger these receptors, right? It's gonna trigger these hair follicle receptors, right? And then so that's why these detect light touch, right? Because when you touch, on the area of the skin and the hair move, a little bit of pressure can already cause, even though you blow some air on back of your hands, right? Which you have a lot of hairs, that you can already feel that pressure. You feel the blow of the air, right? You can test it right now. And because of the very sensitive to light touch. So that's hair follicle. You also have another type of receptors is called Merkel Merkel disc or Meissner corpuscle similarly detect, touch, right? Merkel ending or Merkel disc express a lot on the, lips, for example, area that don't have a lot of hair. Same thing with Meissner's corpuscle, right? Light touch, but expressed in area with, less hair, non-hairy skin. Fingertips, for example. So it allow you to really sense very, detailed information, right? Pacinian corpuscle here. What does it look like to you? Pacinian corpuscle. This area, this end of receptor right here. What do you think it looks like? Oh, some people said it's a fan. What else? It looks like onion. Yeah, right? Because it has different layers here. And that's an easy way to remember also. Onion, Pacinian, right? So Pacinian corpuscle look like onion. And the function of Pacinian corpuscle is detect deep pressure and vibration. So vibration is the keywords, right? So it's expressed in, everywhere, mainly like hand, feet, the place that you basically detect like vibrations, right? Raffini ending is a special one that express deeper in the skin. So basically, if you do the light touch, if you touch your hand lightly, you're not gonna activate the Raffini ending. But if you put the pressure a little bit deep, press on your skin a little bit deeper, then you start activating Raffini ending. Okay, Raffini ending also expressed a lot in the joint as well. So it's buried deeper, detect sustained pressure. Okay, so those are example of skin receptors, right? Now, proprioceptors are the one that, and again, you don't have to worry too much about the details name here, just to show you example, okay? Just make sure that you know like different types. That would be enough, okay? Now, in the for the proprioceptors again is the receptor that detect, basically play a role in detecting position of your body, right? And those are mechanoreceptors expressed in the muscle, in the tendon, and in the joint, right? We will talk about these receptor in detail when we talk about motor system, okay? But you can see that some of these nerve right here wrap around some muscle and then function as the receptors. We have muscle spindle, Golgi tendon organ, and then joint receptors. And joint receptors composed of, so many different receptors together. Some of them are free nerve, some of them are Raffini ending, similar to what's expressed on the deep area of the skin as well. So, right, so different type of receptors. One thing I think, let's see how many slides do we have. Oh, not too much. Okay. I think we can finish the somatosensory system now. Okay, one important functions or property of the receptors is that in your body, throughout your body, you have different amount of the receptors expressed in different areas of your body. Okay, these pictures show you ability of different areas to discriminate or to identify the 2 points. Okay, to understand that, I'm going to turn on my video and then you do some demonstration, and you can do this with me too. So let's see. I turn off the share, right? Let's not do that. Okay. All right, so everyone take any 2 object. It can be like 2 pens. Right here. It can be like 2 pens right here, or 2 pencil, or whatever that have, two sharp object. Or it can be forceps as well. Or if you don't have anything, you can use 2 finger like this. Okay. Now the purpose is for you to try, right? For you to try putting those 2, sharp object. I'm gonna have to put in here, on your fingertips right here. Okay. And you can close your eyes and then feel it. See if you can tell that there are 2 points that are touching your finger. Anyone cannot tell? Let me know how it feel. Can you tell that there are 2 points touching your finger? Okay. Yes, right? Anyone cannot differentiate? I hope no, right? I hope everyone can feel it, right? So even though you don't watch it, you feel that, oh, it's 2 points touching, okay? Now, this is called the ability to discriminate the 2 point, or 2 point discrimination. Okay, so this is one way when the patients come to the clinic, if they have some spinal cord injury or some kind of injury, and you want to test the sensory system, you use the forceps, right? And then you ask the patients, okay, how many points do you feel? Okay, so this is normal result, right? Oh, you can feel that 2 point. Now change from the finger, move this finger to arms, for example. Put it on your arms or your calf, your leg. You can put it on your arms and then close your eyes and see, can you still differentiate between 2 points? Can you still tell that it's 2 points touching your skin? Anyone? No, right? So why is that? Why do you think, that happened? Why your finger, how come your finger can discriminate or identify the 2 points, but the same distance, your arm cannot do that. So it's represented in yes, there's less sensory receptors, okay? Sensory receptors on the arm and more receptors on your fingers, right? So if you see this graph, how to read this graph, the x-axis right here are the distance between 2 points, right? That you are able to tell the difference. For example, here, if you look at the fingers, the 2 points can only be, less than 5 mm apart, and you can already differentiate, right? But if you look at the forearm, those 2 points need to be at least like 35 mm, okay? Or in the other word, if you try again and then you separate that 2 points to be a little bit further, like about 3-4 millimeter, and then you touch it on your arms, now you can start to feel, right? Now you can start to differentiate that it's 2 points, right? So those is, and that's why and the reason for that is because you don't have a lot of receptor like in your finger, right? So the chance that, oh, sorry, I turned it off again. Okay. So, back to this. Hold on. Okay, so that's because you have more receptors on your fingers, right? And less receptors on your forearm, upper arm, shoulder, back, tight calf, right? So that's why you need the 2 points to be super far away in order to separate them. Okay, so that's a general idea for touch. Now, let's talk about pain, right? Sensation of pain. And again, pain is the perception of unpleasant sensations, right? Is it important? Can you live with no pain? No. Why do you think we can live with no pain? I'm trying to like mess around with my slides a little bit, sorry. Okay, so if there's some mutations actually. Exactly. So it's a protective mechanisms, right? So when you feel pain, right? When you, let's say you burn yourself, you move yourself away from that pain, right? So if you don't feel any pain, then you break your arms, for example, you wouldn't do anything because you don't feel pain, right? And then, it will be too late because, you already destroyed the tissue, you did not fix it, right? Or you did not heal the wound, for example. And there's actually some patients who, they have some mutations of the gene that, produce pain receptors, and they actually don't feel pain. And when that happens, it's really bad because, some of them come to the hospital, like, with some of them like step on, some kind of like sharp object, and they're not aware of that. And they come later because, they have some infections or, some further problems already. So that can be bad, right? If you don't have pain. Anyway, how do you detect pain? So there are different types of pain, right? If you classify by location, you have the somatic pain, which is, outside from your skin, for example, or you have visceral pain, right? The pain that comes from internal organ. If you characterize based on property of the pain, you can characterize it into fast pain and slow pain. Okay, so fast pain is those like sharp, pricking, acute pain. Like imagine when you have paper cut, for example, right? Or I don't know, get cut. It's like I don't know in terms and in do you have the word in like Vietnamese? Like how do you say when you get paper cut? What sounds do you make in Vietnamese? Anyone can demonstrate? Let's say like you get like a paper cut. What would you make? What sound would you make? Ouch, like that. In Thai we will call oi. Okay, so in Thai, like Thai people, if you get cut, it's like oi, like that, like oi. And so, those kind of pain, oh, like a sharp pain that come really quickly, right? That's the fast pain. And those fast pain are different from the other pain, slow pain. So after a while, you will notice. So after the paper cut got, better, when you touch it, you still feel pain, but it's a different kind of pain, right? Compared to the first time. It will be more like an aching, slow burning kind of that kind of pain. Okay. And so sometimes also in let's say if you have like a visceral pain, like abdominal pain, usually it can be like those kind of aching pain, right? So slow pain. Those two types of pains are different in terms of the nerve that deliver the signal to the brain. Okay. The fast pain use the nerve with myelin. Okay, so, and I did not mention this in the beginning, but I hope you know or you have heard before that the myelin sheath, what is a property of myelin sheath?
It helps neurotransmission. It accelerates neurotransmission.
Faster. It make neurotransmission. It make the new signal propagate faster, right? So basically, action potential can, instead of just slowly travel along this, it jump, right? Boom, 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 basically. Okay, so that's why the nerve with myelin sheath signal can travel much faster, and that's why you have the fast pain or first pain, right? The second pain on slow pain use the nerve smaller, very small, and no myelin. So it's very slow. Signal goes slower, right? So this is called C fiber for the slow pain, and then A delta fiber for the fast pain. Okay, so this is two different type of the pain. I think I will stop here, and then tomorrow, not tomorrow, next time when we come back, we will talk about the pathway for sensory system. Okay, so I will leave some time for questions. All right, any questions for either the first slides, first lecture, cellular neuroscience, or this somatosensory system that we have. You can ask me now, or you can type in as well, or any questions about the course. This will be a good time to ask as well. Okay, so right, so 40. Okay, so if there is no questions, then I think we will finish here for today, and then we'll keep the rest for next time. And again, I will stay maybe like 5 minutes more for anyone who want to ask questions later. If not, you can leave, and don't forget to email me if you have questions or if you have any requests, right, for the specific topics. And again, don't forget, next time I will do attendance. Okay, make sure that you're here all the times, right? Okay, so I will see you, I believe, next weekend. All right. For now, I'm almost 11 p.m., so I'm gonna go to bed very soon. So you guys have a good day. All right. Eat some pho for me. That's my favorite. I miss that so much. Like authentic. How do you call that? Pho? Pho? The Vietnamese noodle? I can never pronounce it.
Oh, you mean?
Pho, right? Pho, but like PHO, like it's almost like PHO, like a pho, like a Vietnamese
Yes.
noodle. Yeah, that's my favorite. And also oh, you know what? I cannot find anywhere else coffee with yogurt. That's the
You
best.
mean like egg coffee or something like that? Or
Yeah. Well,
oh
yeah, egg coffee is not my favorite, but coffee with yogurt is like the best.
yeah, I remember that.
So have some from me. Okay. All right. I'll see you guys next week. Have a good day.
Have a good day.
Thank you.